Hello and welcome everyone to this session of inoCon webinar series on contamination control strategy. My name is Dr. Subrata Chakraborty. I'm founder and CEO of InnoFar Consulting and Training and InnoVR. Quickly about my company, at InnoFar, we work as a holistic technical support organization for pharmaceutical companies, supporting the industry through consulting, training, and compliance and capability building initiatives. Our training solutions ranges from classroom programs, workshops, and multi-city technical conferences to virtual learning and immersive experiential learning through InnoVR platform, where we develop simulated hands-on training modules for shop floor learning and operational excellence. I will be moderating today's session. At inoCon, our objective is very simple: to create practical, experience-driven knowledge sharing platform where industry professionals can openly discuss real-world challenges, learnings, best practices, and evolving expectations in sterile manufacturing and microbiological controls. Through these initiatives, we aim to bring together experienced practitioners and industry leaders who can share insights that genuinely help strengthen capability building across the pharmaceutical industry. As regulatory expectations around contamination control continue to tighten, organizations across industry are still navigating the practical challenges and implementation and sustainment. Today's discussion is therefore intended to move beyond guidance documents and focus on practical experience from shop floor. We are privileged to have with us two highly experienced industry leaders. Our first speaker would be Dr. Pradipta Kumar Shwai, who is the Chief Operating Officer at Inoxel Life Sciences. Dr. Swai brings extensive leadership experience in sterile manufacturing and aseptic operations, having held senior leadership positions across organizations, including Sun Pharma, Dr. Reddy's Laboratories, Biological E, and Cipla. With deep expertise in aseptic technology, operational excellence, and quality systems, he will be sharing his perspectives on CCS. His topic is CCS myths versus reality. I'm really looking forward to hearing from you, Dr. Pradipta. Following Dr. Pradipta, we will have Mr. Nitish Chakraborty, who is site director at Centrion Pharmaceuticals, with over 3 decades of experience in sterile manufacturing and quality operations. Mr. Chakraborty brings strong practical expertise in aseptic processing, sterility assurance, and regulatory compliance. He will be sharing valuable learnings from media field failure observations, including practical insights from real manufacturing environment. So welcome, Mr. Nitish. We encourage all participants to actively post their questions in the chat throughout the session. We will take up these questions collectively during our interactive panel discussion after both the talks are over. This is a wonderful opportunity to directly interact with our senior industry leaders and gain perspectives on some of the most challenging topics in contamination control and aseptic manufacturing. We sincerely thank both our speakers for taking time to contribute to this knowledge sharing initiative and supporting the broader objective of strengthening industry practices through our knowledge sharing and of your knowledge and experience sharing. So with this, I begin this session. I'm handing it over to Dr. Swai. You can please share your screen and start the presentation. Over to you, Dr. Pradipta.
Thank you, Subrata ji. Just I will share the screen.
Yeah, thank you. Yes, we can see. You can please go ahead.
Oh, perfect. So, namaste everyone, and thank you, Subrata ji and inoCon for providing this opportunity to share our knowledge and views on practical learning of contamination control strategy. So my name is Pradipta Kumar Swai, and thank you all participant for giving your time to this learning session. It is always teach to learn and learn to teach. So looking forward to have a great learning from you as well with your questions. So we will go to the next slide. Sec- uh, So is my screen visible?
Yes, yes, very much.
Okay, so this is a disclaimer. So this presentation will reflect my views, not the company where I am working for. I worked earlier. So the content will be our objective of contamination control strategy and what is the scope, implementation story. There are a lot of story, myths, reality, and learning. So I will be sharing all those with you. Then maturity of contamination control strategy, its management, evolution, and what is the requirement, comprehensive CCS, and the element documentation and information needed. So friends, the contamination control strategy objective lies with effectiveness of all the control design and the procedural, technical, organizational, and monitoring measure manage to identify the risk which are for the product quality and also for the patient and environmental safety. While we want to have the effectiveness, the most important thing to establish an assurance for contamination prevention. And the contamination control strategy basically works for these five areas like microbial contamination, endotoxin or pyrogen contamination, particulate matter contamination, and chemical contamination and cross contamination. So there are a lot of things. These are not new to you. However, when the Annex 1 was published in 2022 and effective from August 2023 was mentioned, so there was a lot of buzz in the industry that contamination control strategy has to be there. And however, you all were doing that earlier. Control strategy name, maybe with the different name like risk based approach, or you want to have a risk identification to prevent contamination, or preventing cross contamination. So all these were in different way you had presented, but now an umbrella document was comprehensively mentioned in the new Annex 1, revised Annex 1 2022, effective from August 2023. So that to drive continual improvement of the manufacturing and control methods to prevent contamination. So friends, this already we discussed the scopes, as you know, in the previous slide. So which is basically focus on various elements of personnel, material, and the surrounding environment. How a contamination can come when while we will go through the different slides, you will be knowing. So you might be thinking that the microbial contamination with respect to the material, or with respect to the environment, or with respect to the HVAC, or all these are microbial contamination. But you will be astonished to see that apart from the material, how while the people came through a bicycle, motorcycle, or a bus or a car, they can bring contamination through that. You will be understanding that later all about how the endotoxin pyrogen can contaminate, particulate matter can contaminate beyond imagination, and which I experienced practically while making contamination control strategy for our organization. So this is it is not only the product, also it has to be patient and safety of the environment, people involved directly, indirectly, and any part of its process which are performed at your site or out of the site, like even the terminal sterilization occurs out of your sites with a gamma irradiation. So what kind of challenges you may face? So that kind of control also has to be in your contamination control strategy document. So there are some myths. We will start with one story. So everybody thought that CCS is a requirement of Annex 1. However, the reality is CCS existed before revision to Annex 1. And this contamination control strategy, which was in Annex 1, is a clarification and more elaboration about the requirement. But that's not the only thing you were doing. We and you all were doing contamination control, and strategies were there earlier in our different documents. So then, as it was in Annex 1, there was a myth that is only applicable for the sterile product or sterile preparation. So actually, friends, this is applicable for all type of doses. From in Annex 1 also, it was clarified. This principally, it is when we talk about endotoxin, generally endotoxin is about the sterile product, not in the non-sterile product. However, the microbial contamination principle, the cross-contamination principle, the particulate matter contamination principle, those all apply to all other dosage forms. So while you are making a CCS, even if your site doesn't manufacture sterile product, you need to work for the contamination control strategy, and for other doses from as applicable, that has to be there as well. So when we understand the CCS and we implemented in the organization with intended purpose, there is a life cycle approach for the CCS and adopting upgradation as and when required. So there are technologies, whatever technologies today it is there, tomorrow it becomes obsolete. So where we stand today with technology, that is tomorrow's starting point. So tomorrow again a new thing will come, and we what we require, science and engineering are like this. Those bring solutions for our challenges, and it is always evolving and always advanced technologies come. So those are when we are adding new formulation, new process, new product. So then also, even if it is advanced technology, we have to assess what kind of contamination control strategy we have be there. So this CCS is a core model which was since decades, and there is a holistic approach for individual process is required. You cannot just mention, yes, we have a strategy. Like I will give example while implementing contamination control strategy, what kind of holistic approach you need to bring. So you have to bifurcate your strategy into people, process, product, facility, equipment, design. So you have to bifurcate all these things. And when it people comes, don't think the people being means the change room, or the people means when they enter the gate. You consider that how people come through a bus. So you can see the picture in on the right. If you see, uh, the bus is on a concrete road, and people are coming out. So their feet will not touch to mud. In the other area, you can see there is the bus is standing on a road where the mud and earth is there. So with their feet, the earthen thing will come inside the company. So these shoes goes to the change room one, primary change room. Irrespective of food cleaning XYZ, the primary change room will be with mods here. And here, as the bus is always alighting in a concrete area, this will not carry the contamination. So you have to understand that also, that how your people are coming, whether they are wearing shoes, wearing chappal. You have a policy of people wearing socks, shoes or not, and then where how they are alighting from a bus, company bus or bike, where they are standing, whether they are transferring any contamination. So what level of thinking, your mindset, and continually improve that. And those kind of assessment required while you are putting a contamination control strategy. That my bus will come, and this is the designated area which is concrete and cleaned every day. And then the people, when they will come down, they will not carry the contamination. Then you will think, okay, bus will stand there, but when they will go home, what will happen? So that also you need to understand, and you have to encourage people that they have to maintain a clean hygiene, and they have to have clean footwear. They have to have a clean practice of maintaining their health, so that they do not bring contamination into your site. So people mindset is very important in contamination control strategy. And basically, who executes contamination control, they are the operator officer which are who are on the floor, subfloor. This is not a document. Contamination control is a mindset, and that it has to be done with a life cycle which never ends, and that has to be with the shop floor operator. So you have to encourage them and bring mindset in them to understand what is contamination control, and they need to follow the procedure for contamination control. So the myth is once documented, you need not to do a further revision because this is already reviewed by consultant. We had done it best. Our U auditor reviewed it and told the best CCS document. So once done, it is not no further revision required. So this is basically a myth. Actually, CCS is to be checked with each and every change control. Remember this. What I told? CCS has to be reviewed with each and every change control. So in your change control SOP, there should be a system that any change control, even if it is a document change control, you are changing a cleaning procedure, you are changing a classification. It is a it is a direct link with the contamination control maybe. But even if you are changing a SOP with respect to a canteen, so SOP or a policy with respect to a canteen, there also your change control should bring whether it has a contamination like a oncology block people going into one canteen and a potent or a sex hormone block people going to another canteen and a general block, how they do not mix up during the process, what kind of controls are there. So those kind of CCS also you need to understand, and with every change control whether CCS is applicable or not, and if applicable, what kind of controls are there. So that is a basic requirement in reality. In every change control you have to do, and it is a mandatory requirement as per the Annex 1 as well. So learning is the change management system has to have a review of impact on CCS for every change control. So if your organization is not doing that, so you can implement that. So there is also another myth that CCS doesn't require to be reviewed during periodic quality management review. So this is a mandatory requirement and must to have the part of periodic quality management review. So you might have the monthly and quarterly quality management review. So what quality management review you conduct, in that there should be few slides mentioning what kind of changes occurred and any impact on the CCS, any cross contamination incident occur, any cleaning validation failure, any cleaning failure, or any microbial excursion, or any endotoxin failure, any sterility failure, any media fill failure, and what kind of kappa or additional proactive actions you have taken, and how you have done organization wide contamination prevention from these learning. So QMR must have the slide of contamination control strategy and any periodic review for any additional steps required to be included. So these are some CCS management evolution. So like understanding it, ensuring all employees grasp the CCS principle and goal. So it is not just training. You ask your people, the best training occurs when you ask people to train. For example, this slide, when we prepare this presentation, I myself learned so many things about the CCS again and again. When I had gone through, I went to shop floor to understand whether the people are understanding it or not, asking question to people. They also ask, yes, I understood or not. One of my colleague prepared this slide, all this presentation. She also learned so many things about CCS. So always ask your employee to train people on CCS, different kind of how you are preventing, how you are doing the change room practices. They have to train. A trainer becomes the best learner. So always the whoever gives the training, he learns 70% thing more than others. And whoever learns by just becoming a participant, he forgets it in next 6 hours. So always ensure all your employees are teachers. So you ask them to teach about the contamination control are in their area about the endotoxin particulate or microbial, what kind of controls you have, what the employees has to follow. So always make people teacher. So that's one of the best way of practical implementation. And then monitoring the effectiveness, evaluating the success of CCS initiative, then upgradation. I will give you a practical example like effectiveness. When we check the effectiveness in one of area, So we were doing fogging, and we started validating that through the biological indicator, a 4-log biological indicator we put, and it failed in the initial qualification. So we understood that the CCS strategy did not work because we put one fogger and we tried to use it for all areas. So let's use more fogger in different areas, and maybe the that will bring more effectiveness. So then we used more fogger together in different areas and segregated also the areas, means for partial fogging one after the other, and we found that all the biological indicator got inactivated, and the qualification was successful. So when you monitor and evaluate things, that will improve it process. And in our in a practical way monitoring, in our every aseptic area door, we put a uh, screen, and in which we mention that how many contamination occurred in last 5 days observation. Means on fifth, 6 days we get the count, and then we put that key. Okay, this is the contamination in which area. So all the operator understand that key, yes, there the contamination are coming, even if no excursion. Even if we get in grade B area, the count, the limit in for a settle plate is not more than 5, and air sampling not more than 10. So even if we get one count, we mention that this location we got one count. So that hits were already known by all the operator or the supervisor who are going inside. So this is one example. There will be hundred of example where you need to monitor and evaluate the your effectiveness of contamination control strategy. So then trending those, when we are monitoring, you have to trend that what kind of contamination you are getting, and how whether it is going increasing or on a decreasing trend. So then adopting new technology, like you can understand the rubber stopper which are there added. Every company who are adding the rubber stopper to the bowl. So you can find in injectable that that's a cumbersome process. Even if you have RTP, you have to open from the inside, and your globe will go over the rubber stopper bowl or pre-feeder. So in practical, there are upgraded technology from where you can open the rubber stopper bowl from outside of the CRAP or outside of the your isolator. So adopting new technology. So these were earlier, it were not there. Since 1 year back, this technology came to few company, and they educated us, and we procured those, and we are implementing those. So those kind of upgradation, you have to adopt new technology and always try to understand the new technology. I will tell all of you to follow a website called farmtech.com. So I don't have any stake in that company, but you must see that they have lot of upgradation and new thing comes every day. That is pharmaceutical engineering website. So it's free for all the people. So you can understand many new technologies. Those are there. It is always every day that site is updated, and whoever becomes a member, they also get a pharmaceutical engineering guide from them. And then a new formulation. So when you are developing, integrating new product, new formulation, you must understand that what kind of contamination challenge. I will tell, we get product of color products, like a product is green, a product is red. So never mix color product, or as a product is having flavor. So never mix flavored color in your area. Suppose there is a peppermint flavor, which is very strong, so that will go everywhere in the dock and everywhere in the area. So how you should dedicate that, or whether you should get rid of those kind of product. So while any product comes, you need to when we have color product, then we always dedicate that contact part, the pipeline, any contact part of a color product must be dedicated. So because there is a chance that it can migrate to other product. And even if with the best way of cleaning, removing all the contact part, you may face challenges. So color product, always there should be dedicated contact parts. So then, while implementation, you should see that across all department, whether they understand and their practices are in aligned with the CCS requirement. Ask people question. The leader should speak last. If you say ki CCS, we have identified CCS and you have to do this CCS. That's not the right way of CCS implementation. Always, while you are making strategies and practice across all department, ask people that what kind of strategy they can bring in, how what kind of new thing they want to add. So always people from the shop floor can bring new idea. Then life cycle management, overseeing the entire CCS process from development to retirement. So from the development itself, you have to check the which are the vendor you are taking. Like for example, I will tell one of the big company who are providing the excipient to all and including all of us as well. So they got a aldehyde content in their product and the product were failing. Many company did not understand that this aldehyde is coming from the vendor itself. The vendor had a problem and they closed their plant and they there was a supply disruption to all for the excipient. So from because all of us we buy the vendor approvals, those vendor approvals maybe 2 years back, 3 years back, where the CCS were not implemented or we did not see. So my view is, as a practical perspective, go to this vendor for all the excipient raw material, critical primary packing material. Check yourself. Don't buy these vendor approvals from RX 360 or anybody. I don't have enmity with RX 360 or any others. But my suggestion, my personal view is the buying the vendor approval through a third party never works, because you understand your contamination control strategy. You must go to the vendor and you have to check what kind of contamination prevention they have and whether they have right control or not. So risk reduction, minimizing potential hazards and safety concerns. So it is not always only the contamination. So there are risks which can bring hazard and safety concern into the product. So that also you need to identify and check. So there is a myth that CCS is applicable to manufacturing site only. In my view, it is not about the manufacturing site only. CCS is applicable to all vendor, material, people, process, facility, equipment, and product involved. And CCS need to be evaluated for new product introduction, which we discussed, a new vendor, material, facility, equipment, and process addition or deletion. So having advanced technology can enhance CCS for sure. This is a myth. Actually, just having advanced technology will not improve CCS. For example, everybody thinks the isolator is best, so we have to take isolator. But if you see whoever has used isolator, you make a process of WIP or water in place for cleaning of the isolator, and that becomes a stagnant area, and you have a coiled handled to use spray jet, which becomes again a area of contamination, because you have unaccessible or recesses which can carry contamination. So how you design your WIP process, how you design your cleaning process of isolator, so that you need to understand how you design your duct cleaning process of isolator. So just having advanced technology will not help. Understanding it, there are so many things for new technologies where you need to adapt the cleaning procedure, like in fluidized bed dryer and other areas. There are cleaning challenges and how those are to be cleaned. That is area of concern. So you need to understand, and with the advanced technology, you need to implement the new area where the concerns are there. So these are CCS requirement. You all know this, so I will not spend much time. Just quickly go through that. Yes, these are associated with design, both plant, process, premises, equipment, personnel, utility, raw material control, including in process, product container closure, vendor approval. We understood that sterilization component, then critical service provider also are to be covered under CCS. So then the process risk assessment you need to do thoroughly. Then all the outsource activity. Many times we outsource many contract sterilization services. We do analysis at different locations. So those have to be controlled, that how you are controlling the contamination. So then process validation, process risk assessment, validation of sterilization, depyrogenation process, preventive maintenance. Preventive maintenance can introduce contamination into your area. So how your plant on plant preventive maintenance standard should be should ensure that no additional risk of contamination, cleaning, disinfection, monitoring system, what kind of monitoring system you have, and those should not introduce contamination. So like you are introducing a monitoring, like I will give a best example here, monitoring system, how they can introduce contamination. So there is a in industry I have seen that we keep all those microbial plate in microbiology area, and then we transfer those plates into the production area. Microbiology is never a very cleaned area, because you have live cultures, you have the contaminations are coming, growth is occurring there, you have isolations there. Your microbiology lab is a risky area. And from there, you are transferring plate to your production area, which is a more cleaner area. So never do that. So in your contamination control strategy, mention that your microbiological plate should be received in production areas. You should have an incubator in the production area. Pre-incubation has to occur in production areas, and then from there it should go to for environmental monitoring purposes that should be used, and then that should come to the microbiology for further incubation and verification. So it should be one way. From microbiology, nothing should go to the production. Even the microbiologists who are for environmental monitoring, they should have an office in the production area, and they should do the environmental monitoring from production. So they should not go to microbiology and then come to the production area. That also your strategy will help in preventing cross contamination and microbial carryover contamination from microbiology lab. So prevention mechanism, you have to trend analysis. We already discussed root cause determination for any contamination or any challenges directly, indirectly involved with that, and then continuous improvement is required. So the comprehensive contamination control strategy requires technical knowledge, process knowledge, risk evaluation, risk mitigation. You must know the regulation. So the Annex 1, you have to mark all those points where the CCS is written, whether you are following that or not. So then guidance, different guidance like best practice document. Even in India, IPA has best practice document on many contamination control. GMP requirement and CCS element. There are many CCS elements are covered under various guidance, including PDA 90 mentions a good thing about how you will implement your CCS. So you can follow all those things. So, friends, I believe we have short time, so I will quickly go through this CCS element. Like whenever you are doing CCS, you should have a concept note for design and facility. Even if it is a existing facility, you don't have the concept note for the design of the facility, prepare a new one. That what your facility, existing facility has, and how your layout, schematic and pressure zonings are helping preventing contamination, how people, raw material, product, waste, water flow, drain flow, design and control, how the set point and criticality of differential pressures are working, your dock diagram, how you are locking the validation after the dock and your lures are fixed, how you are ensuring that you are locking and sealing your lures, and you do not change your uh, differential pressure in different area. Room cascade. So always how the you cannot jump from one grade to suppose D to directly to B grade. You have to D to C and then C to B. So how the cascades are there or not. Then facility finishing, how your finishing to be clean room furniture, how those kind of furniture you should have. Pass boxes, what kind of, whether you should have a sink, you should have a bubble pass box, or you should have one cascade type of pass box. So those you have to define your contamination control strategy. And personnel change room design. So all these you have covered. In premises, equipment, storage area, condition required for material, dispensing equipment, containment system, isolator wrap, alpha beta port, what kind of LF trolley you have, then number of people allowed at one, occupancy control, how you have maintained that, how you know that more people, suppose you are qualifying with 5 people or 4 people, this is applicable for all classified area. So non-classified doesn't have a operational limit. All the classified area like grade C, B, and A zones. So those CB must have the number of people who can be occupied at one given period of time, and those should be controlled. So then the personnel, your induction, GMP training, and rounding process description, hand washing, health check, screening, eye check, visual inspector, overview, clean room, visitor contract management process, how you are doing. Then utilities, your HVAC overview, purified water, WFI overview, process gas overview, then point of use filtration strategy, steam quality control, heating, cooling, and hydraulic system, whether those are coming in contamination. It's like whenever you have a cooling fluid, you cannot take directly cooling water in your manufacturing area. Any leak occurs, that will have the impact. So how your cooling fluids are having impact, and whether you are monitoring their bioload, and whether what kind of cooling fluid you are using in classified area. BSc, TSC verification, then for raw material specification control, movement of pallets in warehouse. Suppose outside pallet you are taking the inside directly, that should be separate pallet. Outside pallet should be separate and inside pallet should be separate. You cannot take outside pallet inside. So how you are doing? Like I will give a practical example how we are doing. We have the color control. Means what are the outside pallet color, what are the inside pallet color, what is the oncology block pallet color, what is the general block palette color. So the palette also, palette movement and palette color will define that it is not getting cross contaminated and not going from one area to other area. Similarly for all the trays, all the gowns, all those kind of movement control you can have to have with the color control. Sampling process, sampling both utensil control, testing and release process control can be done. So there are many other areas. So I'm just skipping few things to save time. So then the vendor approval, then outsource activity we already discussed. So I'm just skipping those. So there is a myth that secondary and tertiary material does not require to be covered under CCS. So actually, secondary and tertiary material can also impact and can leach contamination. Like a label can leach the adhesives into the product, and even the packing metal can smell badly and can leach into the product. So you have to understand this, and secondary tertiary material should be covered under CCS as well. Transport vehicle. So any transport vehicle has to be covered under CCS, because transport vehicle can carry a pesticide, and any fumes can get absorbed into the product, its packaging, and that can be very fatal to the people. So your transport should be qualified, and that should be only for medicinal product, not for any other transport. So the transport should be have a agreement having only medicinal product of which category they can transport. That will be in your GMP agreement. So there is a myth that quality culture has nothing to do with CCS. A robust quality culture promotes contaminant control. In reality, actually, willingness for proactively invest time and resources to improve quality and prevent contamination is a must. And there is always a way that you have to create a culture of following right thing, and that will be directly helping to prevent contamination. So people think management has role in CCS. Actually, management has significant role, and CCS is rightly implemented, continually improved or not. Management has to understand, and always management that appreciates values and contribution skill of employees have more better and stronger support from their employees and subject matter experts. They will be focusing on that. Always networking and learning has nothing to do CCS. Like today you all are here, or we are just talking to suppose I will speak to Subrata or Nitish. how you are controlling this or how you are doing this. So those kind of things you have to maintain a good network and learning from others. You by asking question you never become small. By asking question you are adding knowledge, you are learning from others, and others also learn your views. So always ask, have a strong network for good cross department, cross site communication, cross company communications to understand the best practices and lessons. So CCS is a top-down strategy. Always many people think that actually it's not. Employees to take ownership and quality of product. Those CCS document can be prepared by the corporate office or top people or the site people, but that has to be verified by the operator, and it should be a non-blaming culture for anything, because in reality employees need to understand their responsibility to highlight quality risk, work towards solution, and employees and culture are one of the key element in effective implementation and improvement of CCS. CCS is not about only design. CCS, there is a big role of employees in CCS. So CCS awareness to be created through training. It's a myth. I already told that you need to have make Gemba and ask people to teach you. So that will give you best learnings. So these are some of the learnings. So I borrowed this from PDA Tier 90. So there are governance, oversight, escalation. Then these are, we discussed all these, how we are monitoring control, the validation control, then the contamination control. So you can see this. I will not take much time here. Then personnel awareness, quality culture, quality risk management, and scientific knowledge. So there is, because mostly sterile preparation, next one brings us more focus on CCS towards the operator role. So these are the 7 steps, like qualify operator, identify, train observer, observe aseptic behavior, report to operator and give feedback, improve aseptic behavior. You have to observe their behavior, analyze and train observation, and maintain program. This very few companies do observation of the operator behavior. So you need to have a program to observe operator behavior. This is very, very important. Just not train the microorganism found in the area or environment, particulate matter, or any sterility failure or media fail. You have to observe the operator behavior. That is very, very important from every perspective. So these are, we already discussed this, so I will not spend much time. So this is a way of fishbone, how you should be doing of comprehensive view on factor to have impact on adequate CCI. So we already discussed all those areas. So we are what we repeatedly do, excellence, then not an act, but a habit. So in CCS, this is by Aristotle. So in CCS, that's the same. So you have to do always when nobody is watching, you have to do all those things which are right. So question and answer we will take later. So my special thanks to my colleague Tripti, who made all this presentation and put all inputs and thoughts alive. Thank you all for your kind attention, and my email ID and contact number is mentioned here. Thank you, everyone.
Wonderful presentation, Pradipta. I think there are lots of questions. Overwhelmingly, I'm getting so many questions from the participants. Great presentation. So we will take those questions at the end of our all the presentations. So now I hand it over to Nitish Chakraborty, Site Director of Centrion Pharmaceuticals. Welcome, Nitish. The floor is yours.
Thank you. Thank you, Pradipta, for elucidating a lot of on the CCS, the current topics, what we are all discussing. From here, I take on to the media fields and my experiences on that one. So first, let me set up my yeah. Yes. Is it visible? And I'm
Very
audible.
Much. Yes, yes, perfect.
And it's great to see around 60 persons in this webinar, and a lot of known names are there. Parad is there, then Dishant Kothari, Ambuj is there. So I am seeing them after a lot of gaps. So what I will be trying to do is I will go through a media field presentation, and during the course of that one, I will try to share some experience which may be unique to many of you, or some might have some linkings to that one, maybe have aware of those things. So that will be my your approach in this presentation. So let us begin with that one. Sorry. So first, let me ask everybody over here what we do in this in the pharma companies. There are two parts to it. One is the manufacturing, other part is the sales part. I am not going into the sales part. Let us focus on to the manufacturing side part. As I see, most of the guys are from the manufacturing. So what we do in the manufacturing sites or the manufacturing plants? In my opinion, we produce medicines which are safe for human consumption. This is a very short. So when I talk about those things in that language, what I mean to say is that I don't always preach about the auditors or this or that. We have to preach the people, the operators, the managers, supervisors, even the top management also. Whatever we produce, are they safe for human consumption? So when we are taking a decisions, there are standards given, or the requirements are there in the form of guidelines, and you implement them. When you are implementing, and if you face some issues, what you have to do is you have to ask yourself question: is it safe for the uh, patients? That is what should be our motto. So what is the purpose of the CCS is again to produce a safe medicine. Media fill is a part of the CCS. So when we are doing the media fill, does it ensure the whatever the sterile products what we are manufacturing are safe for human consumption? It's absolutely right. It is the media fill ensures that the your products what we produce in the plant are safe for the human consumption. So what is the media fill? Is the set of all activities which are taking place inside a room. We are converting the sterile components into a product which are sterile or free from any contamination. That is a process called the aseptic processing happens. So this is the media fill, where the facility is there, where the AHU is there, the compressed air, the steam in place, then components, your autoclaves and all these things, sterilization methods, whatever you use, the personnel, they come in inside that aseptic area, and we do the manufacturing, we do the sanitization, everything we do, and we then do the processing to produce a sterile product. So confidence assurance that the product quality, equipment, facility design, component sterilization process, personal training and governing qualification, and aseptic techniques. These are all purpose of the media field to check all these things are okay or not. Through the media field, we do that. Above sessions should be routinely monitored and qualified to ensure the sterility assurance of the product is maintained. Then comes the what is the media field boundary or the scope. That is important. If we say that if it is not a sterile part, and if we include that in our media field, we will be making ourselves life a very hell for that one. So a boundary has to be established. What are the boundaries for the aseptic area? That much we will do. For example, somebody in the terminal sterilization, in my practical experience, I'm now sharing over. I have worked in some company who were doing media fill for the terminal sterilization. So when we questioned about how do you establish what is the limit for your contamination? This we do from where you are getting. There is no requirements established anywhere for this one. So from where you are getting, we are doing this. Who has told it? No, auditors has told them. No, never. So this is my practical experience about the media field doing in the some company which is not a requirement anywhere. I'm talking about the requirement, not about these things, because as per the, if you go to the ISO 9001 2015 QMS, the quality is a fulfillment of the requirements. So I always talk about the fulfillment of the requirements. What is the requirement, and based on that, I work on that one. This is for the suspension process. What is there in suspension process? Two parts are added. One is a vehicle, another one is a product is added aseptically and then produced. The rest of the filling process, and their emulsion are also the same process. Two are different, then pass to a different process, homogenizers and everything, and to get a final product. This is the happens in the. So what are the prerequisites for the media fill? First of all, there should all the equipment should be qualified, the facility should be qualified, the utility should be qualified, sterilization process validation should be over, personal training and gowning qualification should be over, established. All this prerequisites of whatever is written over here. Once they are completed, then only you can move to the media fill. And in media fill, you try to simulate the worst things. That is the longest run size, the fill speed, lowest speed or smallest vial, largest speed, slow speed, like that you do. These are all known, and many times you had gone through all these things, which is you are all aware of these things. So I'm not taking too much time on this one. Important to note is that media fill should not be used to justify practices that pose unnecessary contamination risk. So I was talking earlier that you should not indulge in some activities which are not a requirements, and that creates a pressure on the system and gives a different notations. Which notation I am not trying to make any point over here, but you all understand what is I am trying to say. Again, the PDA-TR 22 also say worst case doesn't mean creation of an artificial condition in an environment which exceed allowed operating condition that should be forced. These were the earlier being bad practices which were earlier being practiced, like the power failure, your pressure drops, the contacting a product on with the gloved hands, and isolated pinholes for the gloves. These were being isolated, simulated, but nowadays they are all now not being done. These are all bad practices. This is all you know. This initial media fill 3 consecutive runs and semi-annual numbers you have to do. Duration should be determined by the largest exposure of media to the environment, sterile component and sterile equipment. Cover all possible interventions, cover operator fatigue and duration of actual service as processing operation. Size of the run, it should be mimic commercial production conditions, cover the representative and types of inherent interventions, at least equal to commercial batch size if commercial batch size is less than 5,000 units, comparable to the commercial batch size if commercial batch size is 5,000. Let me share my experience over here. Usually when we started any new company or somebody into the injectables, the market size is not that big. So we produce hardly 25-40,000 a day, which can be covered under 8 to 10 hours. But with our experience and everything from a previous company, somebody tries to do a media fill for 24 hours. They said we have 120 or 200 interventions to cover all these things. You need a 24 hours to do all these things. So say they try to summon, but that creates pressure on the system and it gives a negative results. So that is very important to understand, to make a realistic approach of how much is your current requirements. It may go on increasing, but it will not increase beyond your capacity. If your size of your machine is 120 vials per minute, if you are doing it for 24 hours, you will not never be able to do that one. There is no point in doing that. It will take many hours, and in my typical experience, if it is a not isolator, if it is a aseptic processing, batch size of around 8 to 10 hours at the most should be good enough to maintain a sterility assurance. Beyond that, it is not manageable for the aseptic process. Why I will say emphatic on that one is that it's all being done by the human beings. If it is not an isolator, human beings don't behave in a very particular fashion. They are very erratic in their behavior. Whatever training you give them, this or that, at the most they will be doing that for a certain period of time. Again, they will go out of way to do something to compromise on the aseptic process, aseptic products. That is the reason my recommendation is always to 8 to 10 hours for a better utilization of the aseptic area, which you will get a better OEE also. Although OE is for 24 hours, but in aseptic conditions, in my word, 10 to 12, even if you go to up to 12 hours, that is also okay. But anything between 8 to 12 hours is good for a daily output. And if you have an isolator, you can go on dragging that to up to your choice, 2 days, 3 days. But how to do the solution for that one, to bringing the solution and changing the tanks and solution, that is also an important factor, needs to be taken into a consideration, and that too should be simulated in the media field. So these are my practical experiences on that one. The fill volume should enough to cover entire internal surfaces of the vials, should allow effective visual inspection of the growth. Headspace should be enough to support the growth of the anaerobic organism. In general, 60 to 80% of the fill volume should be there. Let me share my practical experience on that one. It is said that 60 to 80, even if you fill less than 50%, your agencies does not see it as much of a problem, because in my practical experience, in a 40 ml vial, we have filled vial. Still, they didn't complain or raise any concern on that one, because the process what we have adopted is, after the filling is over, we invert the vials so that your touches all the surfaces, so entire inside surface like the rubber stop or so, and then it is given for a if there is a contamination, it will definitely acquire that one, and growth will be seen observed over the vials. So that is what is again a practical experience what I have gained, and I'm sharing with you over this one. Why we have filled a 15 ml is also because of some constraint. There are the volume what is required to fill more than 50% is not possible for us to manufacture that much of media. So that is the reason why we have decided to do the 15 ml filling and do the risk assessment. Everything was there. So people have your auditors has accepted, inspectors have accepted our risk assessment and gone ahead with the giving the approval to that one. It's not that they have not given, they had given approval. That is the important to note, because I can't give it to the inspectors, but if the inspectors don't accept it, then it is not acceptable. But in my case, it is accepted. That's the reason I am saying it's acceptable. Generally, the SCDM is used for the media fill. Use of anaerobic growth should be considered in special circumstances if it is a anaerobic bacteria, anaerobic conditions, then only the media should support the GPT for the specified organism in a pharmacopoeia and normal environmental conditions. Isolate, sorry. Here are another my experience is that GPT has failed in certain conditions. Post your media fill, when we do the GPT, it is failing. You can understand the conditions where this media were kept or how it was managed. That is the reason why this GPT has failed, and we invalidated the entire media fill. And again, we did that one. There was a long history on that, how all these things has happened, which I'm not in a position to share at this moment of time. but it is for your knowledge that it's very important to manage the dry media in a appropriate manner so that you get a true media fill. Why media fill is important? Because if the media fill is not passing, don't think it as a media fill is failing. The issue is that your product is not safe. So you should raise a concern that if my media fill is failing, my products are not safe for the human consumption. Let us stop over here and think about what is causing this contamination and uh, do the kappa over that and then proceed for that. So people have to raise their voices for if there is a failure. I know we are in Indian culture where in the beginning, when you are being your upbringing is there, people say, don't talk बड़ों के सामने बात मत करो. But that is not the culture in the abroad. They say to anybody bigger than or than us or our uncles, take their names, Mr. Gray, Mr. XYZ. So that kind of culture is there. So they become habituated to a speak up kind of condition. Here, somebody says, our bosses say, don't say anything, then let us do. We continue to do and that one. Nobody is raising concern that this is not safe. So we always should say, give our opinion. If the opinion is not being heard, then it is not your problem. But you should at least for once raise your concern that it is not safe. So that is the, if media feel has a issue, any issue, it can be contamination in the media field, the post GPT. If it is not fulfilling the requirements, you should raise a concern that this is not a product is not safe. We have to look into it. So general guidance, personnel should be qualified once in a year in the media field. Inner gas should not be used. 100% reconciliation of the field wires. These are all the requirements of a media fill. For in any of the guidelines you see, these you will get it. And each and every vial should be identified for time of filling, sequential number, post filling like that. It is there. Incubated for 14 days. That is 7 days in 2025 and another 7 days in 30 to 35. And media fill for the lyophilized should not be frozen and use air instead of nitrogen to break the vacuum. Unsealed containers be exposed for the partial evacuation of the chamber, avoid boiling of media and spill. These are the some comments for media fill for the lyo products. Sampling plan should be enough to capture the possible simulation of the cross contamination. Include aseptic sampling as close as normal production sampling. Before point of sterilization bioburden, after point of sterilization sterility and GPT, extensive EM sampling, continuous EM monitoring during the machine setup, glow monitoring post open door interventions. These are the acceptance criteria for the media fill. In some cases, many of you may be nowadays aware that current requirement is 0. There is no leverage on that one. But when I was in charge of somewhere, we were having, these are the guidelines. This is the your where in practice at that point of time. And this was a new facilities was being erected and we were qualifying that and it was for the US supply. So I hosted the US FDA and I had presented that one of the media fill for one line had one contamination in one out of 3 in one batch. There is one contamination. The inspector noted that one and went out. Went out means after the completion of the audit, he went back to the US. Then I got a mail from him, also a call from him that Nitish, only one contamination in all 3. Yes. Any other? He said a lot of time he tried to inquire this way or that way, but I convinced him that no, it is one. We were honest to note it down, and I have shown you this is the one. Believe me, he accepted that one and given a PAI approval for the product over there. So this is one of the practical experiences I am sharing, that although we all say that it should be zero, and I accept that one, it should be zero nowadays. But at that time, it was prevalent like that, and we captured those things in our records, and we got the approval also. So this is another example where honesty pays. So now coming to our title, process simulation failure investigation. So all possible source should be investigated, like environment, process, procedures, equipment design and control, personnel and their practices, sterilization, validation, utilities, and material. I'm not into my practical experience. I will come to that one later. Let me build the story over here. All, at least the genus and the species level should be identified. Contamination should be compared with the history of identified isolates from all sources, sterility failure, EM failure, excursions, uh, whatever you call them, water, bioburden, etc. So let me share one example of water bioburden in the water. The water storage tank, you might have noticed that there is a vent filter on the top of it, uh, which is again heated to some temperature, say 90 degree, and it has a some shelf life as per the manual of that one. So once we were when we were setting up a one site, we after some time we noticed that the water is failing in the bioburden. After a lot of investigation, say the plant was shut down for at least 20-25 days, then we found that somebody inadvertently opened this filter and found that it has burnt. It has burnt because of the high temperature. The temperature sometimes has gone to 120 also. So the recommended temperature is around 90 or so, something like that. And it also has been mentioned that it should be removed after every 3 months. Later on, some improvement came, and they recommended that now the your literature is saying you can use it for 6 months. Sartorius filter was there. So that is another practical experience. The bioburden can come on that through a different, even TOC was high on that one. So that is TOC was also high sometimes because of your if you have a exhaust of your VHP, and if you're in that floor again you have a water tanks and everything, there is a possibility that your counts will be high because of the TOC exhaust, VHP exhaust nearby. So these are the practical experiences what you may note down to have to check out if you have face some problems or troubleshooting needs are there. The sources of my microbial contamination, as already Pradipta has said a lot of things about that one. But it is generally believed that the people are the most contaminating source for the aseptic processing. Let us not speak about the isolator. I am talking mainly about the conventional aseptic processes. Why people are most contaminating? Because you can see from here, the people generate millions of particles every hour, breathing, talking, hair, skin, body movements, etc. 1.2 million aerobic bacteria per square meter in head and neck region, 0.9 to 3 million square meter on hands and arms, much higher numbers of viable anaerobes. Fully gowned person sitting in a clean room generates about 15,000 particles per minute. So even if you are fully gowned, still if you are in the clean rooms, you generate around 15,000 particles. In them, many can be microbes. Walking person releases 1,57,000 particles per minute. That is the reason why we all recommend that people should not walk unnecessarily. As far as possible, sit in your Peter Baker once was auditing us in Chennai. He saw one of the machine operating for at least for 45 minutes without any interruption, and the person operator sitting on a SS stool. So he was very happy. Well, this is the process where how the machine should operate, because prior to that 2 days he has seen two lines where there is a lot of movement. So this is the reason why the fully people should be sitting and a machine should run smoothly so that you generate a safe product at the end of the processing. Ratio of total particle, that is 0.5, and viable is 600 to 7,000 is to 1. People release around 600 to 1300 total particles per hour in a 0.5 micron size with a 40 CFU viable aerobic organism among these. Properly gowned clean room worker will contribute about 10 to 100 CFU of viable aerobic organism to the environment per hour. These are not mine. This I have taken it from some different source. What is the risk of human contamination? Risk is a function of relation of the human contamination into the dimension of human contamination risk. A gowned operator may release as many as 10,000 CFU per hour or more. It is agreed that the only a significant route of contamination is airborne. Risk source is equal to personal, risk route is equal to airborne dispersion. It is useful to assume that the operators are always contaminated while operating in a aseptic area. If the procedure are viewed with this perspective, those practices which are exposed stable gowning is most difficult to validate. The reliance on the personnel is very high and the associated risk is very high, because human beings are always behavior is erratic. It is not a mechanical like machines. So aseptic technique is again very difficult to validate. The reliance on personnel is very high and associated risk is very high. Similarly, aseptic assembly is on the high risk zone. So what are the desired personal practices? Because see, even if you plan for isolators, there are certain your processes which cannot employ isolators. For example, when you are doing this powder filling, say beta lactam, those which are developed with a high pressure, high vacuum kind of thing, there you cannot employ your isolators. Similarly, when you are doing a vaccine filling, which are live derived from live viruses or bacteria and fungi, you cannot go for a isolator based filling. So you have to keep your aseptic filling also, that you everything going into the isolators. It's not possible in this world at this moment of time. So we have to know about the media field, the people's erratic behavior, keeping all this known there. We have to have sometimes this aseptic filling, because what is the difference between the aseptic filling and the isolator is that in aseptic filling, you maintain area for the grade A and B, and then you bring all the sterilized articles, whether it is air coming through the HEPA, whether the vials are coming through the tunnels, whether the product coming to the 0.2 filtration, the sterilized, everything is sterilized, and then you process under a grade A by the human beings, and then you produce a sterile product or aseptic products. It's a sterile product or aseptic product, whatever you call it. Whereas in the isolators, you set up everything, then you create the aseptic conditions through the VHP. So definitely that is safer for from the microbial contamination point of view. But sometimes the little bit of residues also in the filling area will be cause havoc for the vaccines, or it is not possible because the vacuum pump area, this vacuum pumps which is employed for this powder filling, they break the this your aseptic conditions of the filling area. That's the reason it is not possible in the isolators. So people
we have a quick time check. I'm sorry, Nidish. We have a quick time check.
I will
maybe
quickly
3 to 5 minutes.
So
3 to 5 minutes max.
Yeah.
You can take. Yeah.
Sure. Okay. So I'm not reading all these things. Let me go to the. These are all you know about it. What should be the some facts about the aseptic processing intervention risk? There is nothing perfect intervention. All intervention is a risk. That is what the mool mantra you can say. So every intervention has some risk. So as long as you don't do any intervention, that is the best thing you can do. In order to do that one, what you can do is a gloveless your robotic machines. If you employ, then you can get some kind of. But as of now, these are having a very low speed. So for large volumes, like 400 speed and all, it is not possible at this moment of time. So I'm coming to my do we all agree that the installation of gloveless robotic isolator, single-use system, robotic filling, and will eliminate the media fill failure? In my experience, it is no. I'm sharing media fill failure with the 20% contamination is detected. There was no excursion in the environmental monitoring, no excursion in the personal monitoring. All intervention in the aseptic were free of contamination, as we have noted down everything. Genus species of the contamination was identified. The pattern of the contamination was noted. It was found that the contamination is basically into the when it initial vials, after the lunch, after the dinner, like that. Whenever there is a longer pauses or the residence time of the vials in the entire belt is there. So this media fill was for the initial qualification of the. So the leak test of the HEPA were fresh. Around a month back only it was done. So we never doubted on that one. But it has to be checked again for as per the procedure. In order to find out the source of the microbial contamination, entire plant is mapped. As Padita was saying, showing some photograph that food in a mud or in a concrete one. That has really a value over here. So we mapped the canteen and everything was mapped from where it is coming. We mapped the canteen because people are first of all go to that place to have a tea and then come to the production area for operation. The micros of the component preparation, that is vial washer and tunnel, got mapped the micros of the tunnel was having facility of sterilization of cool zone. The leak test of the cool zone HEPA were done a month back and the result was satisfying. Just for your information, the frames of the HEPA are usually aluminum, whether it is a room HEPA or any other LAF HEPA, but the frames of the high temperature resistant HEPA are of stainless steel. The reason, the coefficient of expansion of the SS and the cement holding the HEPA are the same. So there is no uneven expansion contraction, thereby no cracking. The leak test of the HEPA of the diaphragm was done again and found failing. The HEPA of the cool zone was taken out for verification, found damaged. Refer to the next slide. You can see how it was damaged. The damage were investigators. There was no intervention in the cool zone after the last leak test, as per the interview with the operators and the validation team. After a lot of brainstorming, it was concluded that while removing the probes from the cool zone after the thermal validation and the HEPA filter verification, operators has unintentionally damaged the HEPA filters of the cool zone. So this is the, if you see this area, these are all damaged. This stainless steel are your frames of the tunnel, but behind this one, if you see, that will be whenever you get an opportunity to look into the HEPA filters of the tunnel, take it out and see, you will find that the high temperature resistant wear of stainless steel, because that is a requirement by the manufacturer. So this is my personal experience that can happen. So what was if there is a gloveless your isolator with a media fill would have failed. What is the cause of failure? The HEPA of the cool zone of the depressurization tunnel were leaked. When the belt of the tunnel has paused, residence time of the valves was high compared to the regular moving valves, thereby getting contaminated. This has resulted in the media fill failure. there are a lot of remediation can be there. I'm not, what we have at that time done is we put up a policy that after the completion of the leak test, your qualification, thermal qualification and everything, we will do the your leak test, that is the DOP testing of the HEPA filters. So that is my one of the big experience, because this has led to a 6 months delay in the qualification of the line. So there was a has a big impact on our business and everything. So from my practical experience, if you can learn, sometimes get this one, it will be better for you, so that you lead time is or remediation time is shortened. Thank you. We can take up your questions.
Thank you very much, Nitish, for wonderful presentation. Now it is time for answering the questions. There we have lots of questions. I will try to kind of collate these questions and pass on to you one by one. So the first question, it is to Pradipta though, but Nitish, you could also share your thoughts on this if you want to, and vice versa it would be for the questions that has come to you. So during each CRF review for the impact on CCS evaluation, is it required that CCS should be revised for each case or can be revised at certain frequency?
Yeah, this it's a very good question. Actually, in each change control, everybody has an understanding CCS is a document which needs a revision of the strategy. Actually, no, you have to add addendums. Like suppose a new process came and the process itself has certain thing you need to add. So you have to add that based on the new process added. These are the strategy added into our contamination control, and that addendum has to be added. And in your previous original CCS, you should mention that any revision to process, any new facility, any new addition of the line, the addendum has to go based on the CRF change request form. The addendum has to define what kind of contamination control strategy approach to be followed. And after implementation of the contamination control strategy, you have to check that whether those were implemented rightly or not, and the section of the procedure, like in your SOP you are defining, or in your protocol you are defining that where how you are going to control that, or in a cleaning validation you are defining for a new product introduction, it becomes the worst case, or a new cleaning chain. So how you are going to do that? So all these particular point 4.1.2 of protocol number so and so, version number so and so mentions this, and any revision to this protocol in future, these hashtags should not be changed. That means your strategy, which are fixed for particular challenges, those should be hashtagged, and in the end document you have to mention that these hashtags should not be changed. This is the base for this contamination control strategy. And in that case, if you are mentioning these hashtags should not be changed, in case a change required to that, a justification note has to be provided. So those kind of things will help preventing every revision of your contamination control strategy. Thank you.
To make it a little, I will add on that one. See, in my opinion, you can have a certain frequency of revision of the CCS, but you, as Pradipta was, I am also in the same platform, that whenever you are changing or change control is there, or any requirement is there for a revision, or you can add addendum and keep all this addendum and revise at a certain frequency, say 6 months or something like that. So this is where also can be done.
Yeah. So the next question is, what are the key aspects shall be ensured during change review, evaluation of site, procedure, process change with regard to CCS sterility assurance?
So the key things are microbiological. If you are talking about sterility assurance, so what kind of microbiological contamination and what kind of particulate contamination. Microbiology doesn't mean only microbiology, because microbiology is directly correlated to the particulate contamination, because microorganism doesn't have legs, they cannot crawl or they cannot swing. So they need a particle to move. Particle is their vehicle to reach into a substrate or a product or in process contact parts. So how you are doing the particulate contamination, how you are doing the microbial contamination, and when you are talking about the sterility assurance, apart from this, as Nitish mentioned earlier, people are the largest source of contamination. So how you are preventing people led operations and preventing lesser people or lesser intervention into that process, that you need to check as well. And any sterility assurance, it doesn't just need these three, it needs more actions, like how your process are closed transfer system, how you are using the techniques, what are the advanced barrier system you are using, advanced technique you are using, or any single use system you are using or not, and how you have designed your equipment and system to prevent intervention into your fast air, and how you are getting online information on your air velocities, because air velocities is one of the most important factor which people fail to understand, because your smoke study, your recovery, your sweeping away of particle or contamination, all depends upon the air velocity. So how you are monitoring your air velocity continually for each zone. So that will help. How you are monitoring your pressure differential for each zone will help. So you have to, it's a long, the answer to this question is very long, but to certain this, you need a holistic review. So the some few points I have added only.
That's a good point. In fact, just I want to add something. So when you make your initial CCS, you know what change makes what impact. So now, as you go on, there are deviations, there are changes happening day in, day out. You know what might have changed. So your assessment becomes easy. So I mean, trying to help you. Of course, I agree to what Pradipta said, but I'm trying to help you how to look at what to look for. Okay. Yeah. So the next question I think is related to how, if there are multiple changes with regard to CCS, what is the preferable frequency? I think we spoke about frequency already. Uh, you know, uh, what matters for you. You have to understand what changes could impact. So that's what Pradipta just now said. For a new facility, what are the critical aspects shall be ensured with regard to CCS? Do you want to take this? What are the critical aspects shall be ensured for a new facility with regard to CCS?
Yeah, that was exactly in the presentation. So it covers everything, like your layout, your people movement, your utilities, your, like for example, you might have one air compressor and you are supplying to two block. So, or the air compression system is one, and you are just providing, you are telling, no, this is only for the equipment air. So having a common is no impact. But that can have a backflow from a oncology block to other block, and that is when there is a breakdown, and how it can take the contamination from oncology to the general if the air compression system is same, whether NRV are there or not. So from utilities, facilities, layout to people movement, material movement, your like where to keep your water tank, whether it is underground you want to do or you want to do a overhead tank for the water. Generally in your lifetime you have seen the underground tanks. So for in CCS you have to define what is the benefit of underground tank. Any leakage occur in the underground tank from the adjacent, you will be knowing later after identifying that where the water is already used. Suppose you will make it on the earth, so that the load is huge and the capex will be more, and whether you want to do that to prevent contamination. So a lot of things in a new facility. So what are your learnings from the other areas, other facilities, and also from design stage to HVAC? Like I will tell one important thing, most of you might be knowing or few may not be knowing that in a new facility. Whenever you are providing LF in a sterile facility, ensure its input air is from the ducts, like the supply duct of the room, or even you can take from the return duct of the room, no matter, it doesn't matter. But the LF input air should not be taken directly from the room. That will, whenever you will open your door, the rigid partition of that LF, or you are working on LF, that will short circuit. The air will short circuit. So that is one important design you need to always remember in a new facilities, because older facility always LF you will see the input air from the side of the LF. So never do that. No LF has to have any room air suction from its side. It should be always from the top. So that is one example. There are many. These are short time. You need to have full day or 2 days time to discuss all these questions. There are many things you need to learn in a new facility.
Absolutely.
Those are some few
Absolutely.
examples.
There's another important question. I mean, very good question. How do organizations typically manage CCS across multiple facilities? Is it better to maintain separate CCS documents for each site, or sorry,
Yep.
Is it better to maintain separate CCS document for each site, or a single integrated CCS covering all facilities?
It's a wonderful question, friends. If you understand CCS, it is not organization specific. CCS is process step specific. Means your one site's process, or one block process, or one equipment process, or one room process will be different than the other. So your CCS guidance document can be a corporate one, that how your CCS flow to be, how your CCS governance to be. That can be a common for across organization. The governance mechanism or the guidance mechanism should be corporate one. But the actual CCS should be specific to process material. Even each material you have to do so CCS, like excipient. Suppose you are doing polysorbate or polyethylene glycol, you are getting those kind of excipient. Whether DGEG is performed on those, each container DGEG is performed or not. So what kind of requirement are there? Suppose you are using purified water for say oral solution, whether PCC checks are there or not. So in your strategy, you have to mention. So those are specific to your dosage form, specific to your process. However, the governance can be a organization specific across all site. That can be a central or corporate driven, but the actual CCS has to be made from the subfloor or from the vendors, from the service provider, specific to that process and specific to those in process control and specific to transportation requirement. Those should be specific to the site. It cannot be a common one. Thank you.
Yeah, risk assessment for the every site is different. Every site is different, so risk assessment will be a different. That's the reason I fully endorse what Pradipta says. Risk, your CCS has to be a different for every site. It cannot, of course, the guidance as this is can be a corporate driven, but when it comes to actual doing the CCS, it has to be a separate for each and every site. I think you are on mute.
You are on mute.
Subrata.
Yeah, Subrataji.
Oh, I'm sorry. The next question is, in sterile API manufacturing, dissolution, crystallization, and drying activities in closed sterile systems, background area is ISO 7. This area is big and having many pipelines. How to clean pipeline outside of ISO 7?
Yeah, see, ISO 7 is a clean room. I believe ISO 7 you mean to grade C because always the classifications are in rest condition. So grade B in operation is ISO 7. But what you mentioned in a sterile API facility in a closed transport system, generally grade C is absolutely fine. So in those cases, in clean room, which is grade C as well, there should not be any uncleanable recesses. So any pipeline that I know I have seen this API facility because I work for API facility also. So where those kind of external, you need not to worry much. Those are like sleeves cleaner. Sleeves cleaner means in a pipeline you have to have half crescent type cross. So where you have to clip it always from the top bottom, and you have to just run through it. So if you do that, then it will get cleaned, and you can have your frequency whenever there is product campaign is going on. You may not clean the outside one for a single product for 1 week time, and you can weekly basis clean those by external pipelines if the same product is going on. Internally, you have to anyway clean those. But there are ways you have to find out, and you have to design in such a way that it should be easily cleanable and there is no uncleanable recesses. It is a CFR requirement. It is a crime if you do not have easy cleaning facility. You have to made such design in API facility for sterile API where there should not be any uncleanable recesses. But of course, there are different methodology which you can clean, as I mentioned, the sleeves for cleaning of pipelines.
Nitish, do you want to add anything on this?
No, I'm not
Great.
much conversant with the API.
Okay, the next question is for the new sterile facility. At what stage we need to develop CCS document? I think this is again a very important question. I see day in, day out such questions coming out from many people. What about preparing a CCS SOP initially to cover the topics and to define CCS related actions in facility change controls, CCS related actions, and after project completion, final document can be drafted and signed off? Or secondly, if we can keep a CCS document as draft and leave from start of the project to cover and complete till project end all CCS related actions. Yeah.
This is a very, very important point. Whoever has asked question, thank you to him or her. So friend, if you understand CCS, your strategy after making a plant, existing plant, you have to write down with the available plant design, available process or available procedure, and then add or modify the process. When you are making a new plant, you have all the scopes ahead. So you have to understand this plant is meant for what. So there comes a concept paper. So your first document for any project is concept paper. In my presentation, I have covered this. So your first document should be a concept paper that this plant is meant for what. It's a biologic plant, or it's a potent block, or it's a sex hormonal block, or it is a steroid block, or it is for what you are minting for. And then you have to mention that what kind of challenges you may face. So the position paper or concept paper should be made before the your field is green. So that time the concept paper has to be made, and then the CCS document has to be made after the concept paper before you order or you finalize your layout. So, because in your concept paper, whatever you are mentioning, that should be transferred into a detailed CCS, and that CCS will be transferred into your layout, your design. So, always the CCS to be the first document attached to the change control, because change control will be there. Suppose your new plant is there. Earlier in when 25 years back or 30 years back, we have started at that time plant were made, and after making we were having change control because we don't have QMS. Now this is not acceptable. You're fast to hire any greenfield. You have to hire your quality system, quality people fast. You have to put your concept paper and change control fast. Maybe to be manual change control. You have to have that. Your each equipment, each layout, each digging has to have your change control fast with your water system, your whatever you are doing. Your change control is the fast document. So those earlier days and our old guardians who were there, they will tell, अरे यार पहले कुछ भी नहीं था, हम ऐसे ही चलाते थे, करो यार तुम्हारा A4 साइज पेपर के लिए प्लांट रुका के रखा है. No, that's not the way. Now the mindset has to change, and CCS has to be prepared after the concept paper before you start digging of the plant making. So hope I'm clear
Absolutely.
to you.
Absolutely, I fully agree. And I would rather also say, I also have experience, Pradipta, where a bad process design, say, for example, where in an aseptic manufacturing, compounding activity, the R&D or the technology transfer guys would expect you to collect sample from the bottom valve of the manufacturing vessel, which is highly risky in an aseptic environment. So that means
Not acceptable. Yeah.
your CCS should start from there, not after conceptualizing your plant, rather during conceptualization of your product, the process development. So now it is irrespective. I mean, there is no worry when you say that I have a CCS document, I signed it off, and I revise it again. You keep it revising, not a problem. We always said that CCS is a live document, you keep on revising. But don't worry about those formalities. You must have CCS from the time you start thinking about a product. Okay, now let me take quickly a
just
few
one
questions.
thing.
Yeah,
I would
please.
like to add one thing.
Yes.
See, as we all understand that CCS is a dynamic document, point one. See, I've been very categorically told by the US FDA that change control has to start from after the qualification, IQ/OQ is done, not before that. This is my
Nitish. So no, I understand. See, that's the view change. FDA cannot teach us because recent FDA inspector, whoever initial people joined this, they will with me. Recent FDA inspection was in inoxel. The inspector told that you have a right process. The change control is raised prior to every orderings, prior to PO, not qualification. That is a must. And if you are doing after IQ change control, it is a total breach of quality because you cannot. Your introduction has to have a change control. Na? Your order has to have a change control. Your URS, you cannot prepare a URS unless you have a change control. So that's US FDA cannot run our facility. So if they are telling something
right.
it is incorrect, yeah. I
there are
have a different
different ways.
view.
There is no hard and fast rule about that. What I'm trying to make a point. There is no hard and fast rule on that one. No statutory requirements or regulatory requirements on that one. The point here is there are people do a different way. Point number one is some people do a concept note, then they technical specifications, then all this URS and everything comes up after that. And once you do that, because you are not aware of the things at the very beginning, you do the risk assessment based on that, you do the requirements, understanding, you give the orders based on that. During the order manufacturing, there may be a certain issues on that one, so make a little bit here and there correction also. So that is the reason why a practical approach, as when I was doing the presentation to the US FDA, it is not about they have come here. I was there in their office in US, Springfield, where I was interacting with them for the around 14, 15 inspectors. They were sharing that this is the what should be done in the water. They have given a lot of inputs for our plants and all these things. I'm talking about it is they are also said it's not necessary that you should have this cover only. So there is no laid out.
Yeah, of course.
Yeah,
so wonderful discussion. I'm loving to having this discussion, but due to paucity of time, let me take one or two questions from the media field side. I find some of them very interesting. What is the criteria for personal qualification through media field? And is it necessary that the personnel required qualified on each and every intervention? This is a very good question. Again, lot of confusion with regard to this.
See, the requirement is whatever interventions you are going to do, you have to say you participate on those interventions. Within the 6 months period, you have to, or 1 year period, sorry, not for 6 months, once in a year, you have for first 6 months, some operators, and next 6 months, another few operators, they can critical things. For example, opening, assembling, everything. For suppose you have 6 operators, the first 3 can first 6 months, and the another 3 can be in the next semester. They can do it. So that is the requirement for the operator qualification through the media field. But if you're somebody is your helper in aseptic, you want to qualify him also, so he has to undergo some training, and then only you can qualify him. It's just a matter of you come gowning qualification, tomorrow onwards you are doing the media, you are assembling also. That is a troublesome you are calling for.
Yeah, I
Pradipta,
would
do you
like to
want to
add a very important aspect here. Very, very important. Friends, whoever working in sterile area, you need to be qualified in each and every intervention. The word is, your question was whether each and every intervention, answer is yes. You have to. The person who is assembling on the left side of the equipment at globe port number 22, he has to do that much only. He cannot move to the other side of the assembly and do the globe port number 23. So it is so stringent. Your qualification should be specific to the aseptic intervention. If you are doing any vial removal from the turntable, then you are qualified for vial removal in the turntable. The other operator qualified for the rubber stopper removal is not qualified for the turntable remover. In old days, we were grouping these actions. Like group A is assembly, group B is intervention of the fallen bile removal, and group 3 is aseptic connection. So they like this. And now it is not acceptable at all, because people to people will vary. The person has to get qualified in particular intervention. You please read 483 and warning letter of Sun Pharma. So which was most recent, and you will find all these that your operator were not qualified on each and every interventions. So it is a must, and wherever I worked, I had ensured that this is done in Dr. Reddy's, in Son, and in our inoxel as well. So it is the most that operator has to go through each and every intervention. Prior to they do that. Thank you.
Great. Thank you very much for this wonderful discussion. So we are not able to address all the questions, but to all the participants, if you can keep on posting. So we have a wonderful platform, inoCon. It's free. Please connect to the LinkedIn channel, and you can keep posting your questions, and expert like this can always keep on responding or interacting with you on this platform. So with this, I want to thank both the speakers, Nitish and Pradipta. Thank you for very much for your wonderful presentation, and thank you everyone who participated in this program. And you look forward to many such programs in coming days. Thank you so much. Have a great evening.
Thanks
Bye-bye.
all to the
Thank you,
attendees for
everyone.
their patience hearing.
Thank you.
Thank you. Thank you.
Bye-bye.
Bye-bye.
Thank you.
Bye-bye. Bye.
Bye-bye.